If you're a bioinformatics researcher, then managing vast amounts of datasets is certainly part of your daily business. But we all know how time-consuming it can be to set up a new analysis, and particularly if we have to curate our data sources. So we will take a look at the ReadStore database, which is a tool designed for data scientists to manage their omics datasets, thereby reducing the time required for analysis. In particular, this tutorial will focus on the Python client and explain how we can simplify bioinformatics scripts written in Python using the ReadStore integration. I'm going to walk you through 3 principal use cases that hopefully inspire you to upgrade your analysis as well. So let's get started. We obviously need to install the PyReadStore client in order to get the package imported in our Python scripts. However, this is super easy since it's available in the Python package index, and as such we can install it via Python pip. Depending on your setup, you may actually want to install it within a Conda environment or virtual environment to keep things consistent. More information on this you can find in the docs, which I'm linking below. By the way, the client is open source, so you can extend or modify it, embed it in your software, however it best suits your automation purposes. And once we completed the install, we can switch to our Jupyter notebook and see if the import works. And when we run this import statement, import PyReadStore as rs, we can see that this works, which is great, so we can continue to the next step. Okay, now in principle we're ready to build our bioinformatics workflow, but of course we need data to work with. And since we're already working with the ReadStore Python client, we should also get our data from the ReadStore database. So moving to our ReadStore web app, we can first check out the projects view. Projects are groupings of datasets and associated metadata, and this comes in very handy if we want to automate our pipeline, since we can build our data analysis around those projects. We have one project here named scRNA-Seq, which holds datasets that compare a tumor versus an adjacent normal tissue and investigates the gene expression. And if we want to get more information, we can simply scroll down. We see the project name and the project ID, and we also see associated metadata. So as Bioinformatics Pro, it's really important to keep the metadata consistent and build consistent annotations. And for this reason, you can actually store your metadata along with your projects and datasets in the ReadStore app. And here these metadata sets come in as key value pairs, so like a JSON dictionary. And this is really easy to flexibly build categorical annotations. But where's our actual dataset stored? For this, we go to the Datasets tab, where we get an overview of all the datasets that we have on our app. And we can see that for our scRNA-Seq, we have 6 datasets, each 3 replicates for the tumor and the normal tissue. And if we toggle here the metadata view, we can get an overview of all the metadata categories like the tissue and the replicate that we have stored here. This is what I was just talking about, building these consistent metadata annotations for each dataset. And if we want to get access to our actual raw data or process data sets that relate to these data, then we can select the data set, and here in the details view we get access to the FASTQ files for individual reads, and we can move to the Pro Data tab, which holds process data sets in these count files for individual bioinformatic pipelines, which we've stored here along with our data sets and raw data and metadata in our ReadStore app. Now back in our Jupyter notebook, we want to make use of the ReadStore client. But how can we actually interact with these projects? So here's what we do first. We will first create a ReadStore client. This ReadStore client contains all the authentication for the backend. This is super easy. We can simply create an instance. You can also set explicit usernames or tokens, and more information on this you can find in the docs. Now let's see if we find our cancer projects. For this we run the list projects command, and what we get is an overview in form of a pandas data frame that contains all the projects, as we see our scRNA-Seq projects along with metadata and some more information. Alternatively, we can also run the getProject command, where we specify an explicit project name or ID and get some more detailed information in form of a pandas series. Finally, we can also create a new and empty project. For this, we initialize it with a new name and description, along with some metadata that we want to set. And if we run this command and move back to our data app, we should see that we have new projects popping up here. Now this first part we want to run a quick analysis and check how many reads are found in each FASTQ file of our datasets. For this, we will just count the lines that we find in each FASTQ file and then divide this number by 4, which gives us the number of reads. And we have the small function prepared here that is called countReads, which takes the FASTQ file path as input, then opens the gzipped FASTQ files, and counts the lines in each of these files and divides the result by 4, and that gives us then the number of reads. And this little function we want to now automatically apply to all our FASTQ files that we stored in our ReadStore database. But first we need to get our FASTQ files somehow, so the ReadStore Basic version stores those local file paths. And if we obtain the datasets from our datasets, we should get access to the actual file path on our system. For this, we start by listing all the datasets that we find. We can specify a project name here. And if we run this command, we get an overview of those 6 datasets that we already saw in our overview. Now, similar to the project get command, we also have a get command here that gives us a more detailed overview if we specify a specific dataset name, in this case the tumor replicate 1. And here we get some more detailed information on this one in form of a pandas series. Still, we don't have our FASTQ files here, so what we need to do is actually call the getFASTQ command, which will retrieve the FASTQ files for a specific datasets if there are any uploaded. And here we will do this now for this tumor replicate 1 sample. And if we execute this command, we again get a data frame here. And in this data frame, we get some more information specifically for the FASTQ files, because ReadStore internally, when we upload datasets, performs a QC check on these data. So here we find, for instance, information on the read length and already the number of reads. So we can use this to compare our datasets later. And with this command, we can now also extract here the upload path, which corresponds to the file path on our system. And this upload path we can now feed into our function if we move down a little bit here. So what we do here is obtain all those FASTQ files, and then for the first upload path that we have, we're feeding the path into our countReads function, and that should give us a number of reads if we execute this. And as you can see, once a function finished, we get the number of reads, and this number of reads actually corresponds to what we already have in our database. Now in the next part, we will load some process datasets and perform a small analysis using the metadata schema that we just defined. But first, what is meant by process data? So process data are all types of files and annotations, formats that are the result of processing our input sequencing files. Those can, for instance, include gene count tables, gene expression matrices, alignment files, or more complex downstream analysis results. And for a pro bioinformatics researcher, it is of course essential to be able to access those files instantly without having to search through folders or directories, where chances are high that we will actually lose track of those files. And as we saw in the beginning, we can attach process datasets to the datasets along with the metadata and the raw data, so that we keep everything bundled and that we can access them through our Python client. And that is what we're going to do now. Okay, so we're back in our notebook where we can run the list pro data command. And here we specify the project name, which is our cancer RNA-Seq project, along with the data type of process data that we want to return. We've seen before that this is counts TSV, so gene count files that come from a gene quantification algorithm. And if we run this command and have a look at the results, we see that we get for each dataset, we can see this here by the dataset IDs or names, we get one gene count file that comes from the software Callisto. And here we also get the upload path, so the file path where this file lives on our file system and how we can access this. And what we are doing now is that we want to load all those gene count files, combine them, merge them with our metadata, and then make a little plot, a short analysis to illustrate the workflow, how we can go from our process data sets via our metadata through an analysis. So first step here is to read the individual gene count files. For this we use pandas. and then perform a concatenation. So we combine all those individual data sets here. And then in the next step, what we're doing here is we are obtaining the metadata column from our dataset list, and then we combine those into a single data frame. And this gives us, we can illustrate this here. This gives us an overview of the metadata for each individual data set. So that is super easy. This takes 3 lines of code to get a consistent overview of the metadata that we have for each of the data sets. In the next step, we can merge this now with our gene counts so that each data point is annotated with the right type of metadata. And once we do this, we have a summary table, and the summary table we can now use to perform some exploratory analysis on. So here, for instance, what we are just doing is for the GAPDH gene, we're going to plot the tissue, which is one of the metadata variables that we loaded in, along with the TPM, so an expression, a gene expression estimate, and we're going to compare this across those categories. And when we create this bar plot, we get an overview of the gene counts here and the error bars that come from the replicate. And this is just to illustrate how easy it is to set up these analyses that rely on having access to the categorical metadata variables through the Readstore API. It's basically always the same schema, so this type of analysis can also be done for differential gene expression or any type of bioinformatic workflow that you can imagine. It's very easy to automate this building around the Readstore Python client. So we've seen now how we can access process data sets using the Python client and build a downstream analysis on top of this. The key advantage of this approach is that it's easily scalable because your team can manage all the metadata in the Readstore app, and this gives you very consistent and flexible access to all the data you need for your analysis. Finally, what we want to talk about is how we can upload process data sets. Of course, sometimes our script may produce analysis files which are very large, or we want to store these results for another type of analysis. And just writing them to disk always leaves the risk that we don't find them anymore or that it's not trackable. But also here, we can simply create a new dataset and upload a downstream analysis or aggregated results as process data. And that's what we're going to do now. So back in our notebook, the first thing we're going to do is to create an empty dataset, which we are calling cancer RNA-Seq results. So this has no input raw data attached, but we can use it to store our aggregated result files. So for this, we are also attaching this. We are defining a project name here, and this will basically make this data set attached to the cancer RNA-Seq project that we saw in the beginning. And if we execute this, we create the data set. And now the next thing we do is actually save those aggregated gene counts that contain the metadata to our disk. So we define a file path here and then write them to a CSV file using pandas so that we have stored the file on disk. And then in the next step, we actually prepare our upload command. So this upload command requires us to set a name for the pro data set, the actual file path, and a data type, which we just call result here. So we're free to choose whatever we want. And then, of course, the dataset name that we want to attach this dataset to. And this is, in this case, the cancer RNA-Seq results that we just created. And if we run this command, we perform the upload. And what the Readstore server will do in the background is actually make sure that all the file paths that are registered there actually exist, so that there can be no inconsistencies between what we store on disk and what we store in our database. Now let's move to our Readstore app, go to the dataset section. We should see that we have a new dataset popping up here. And we can see that we have our RNA-Seq results here. And if we move to the process data, we see that we have our gene counts here. And similarly, we can access them back again. And that was the upload section. So in summary, we've seen how we can easily upload result files or process data back to our Readstore database so that we can share them with our team. We were initially setting out to explore how, as top bioinformatics researchers, we can speed up our data analysis and workflows while keeping track of all our datasets, files, and metadata. We looked at 3 case studies where we first loaded some raw sequencing files directly via the Python client. Second, we loaded some process datasets that contain gene counts that we merged with our metadata annotations to drive a downstream analysis process. And third, we saw how we can upload aggregated results to a new dataset, and that all fully automatic through the Python client. If you have questions or comments, you can leave them here. Readstore is available as a free demo version for you to try on the website, which I'm linking below. And otherwise, I wish you a great day.